President Fidel Ramos. The award-winning National Corn-Based Farmer Scientist RDE Training Program, or FSTP, was also established around this time and still remains as one of the university's primary extension programs. In the field of instruction, the book titled Integrating Sustainable Agriculture in the BSA Curriculum, the UPLB College of Agriculture Experience, was published in 1999. At present, the College of Agriculture and Food Science, or CAFS, continues to be the premier institution of higher learning in agriculture and food science, and remains committed to nation building through its instruction, research, and extension programs. In order to produce the best graduates, all of our courses are outcomes-based education, and continuous improvement of the curriculum is being practiced to meet the challenges of the 21st century. As testament of excellence, CAFS is constantly recognized by different organizations. In research, we continue to implement relevant studies with focus on the 12 priority commodities to address food security. Moreover, technologies are constantly developed to aid in responsible food production. Supplementing these are extension activities to disseminate the knowledge generated to improve food productivity and livelihood of the people. We believe that present technologies will future-proof our food systems. Some of our notable developments are ID-Tech, Coraginam, Plant Health and Pest Clinic, Pest Management Tools and Techniques, Smarter Pest Identification Technology or SpeedTech, Snap Hydroponics, Drone Technology, Rapid Immunofilter Assay for Virus Indexing, Nanofertilizers Fertigrow, Orbogrow, Nanobiosensors, Biochar. We at the College of Agriculture and Food Science will continue to promote and nurture a culture with innovative yet conscientious minds and cutting-edge skills to lead and serve the country and the region. We are one college with one goal towards sustainable food systems.
The problems, pest invasions and outbreaks, crops diseases, massive use of pesticides, food security policies, confusing field practices. While Philippine agriculture has been a major arm of the government in achieving its long-term goal of food sufficiency, its objectives can only be attained fully in partnership with a strong pest management institution. The answer: the National Crop Protection Center. The National Crop Protection Center. The remnants of Rodent Research Center in 1968 paved the way for the establishment of the National Crop Protection Center on May 19, 1976, by virtue of Presidential Decree 936. Its founder, Dr. Fernando F. Sanchez Sr., exerted all efforts in expanding the rodent studies into more comprehensive pest management programs by forming teams of experts in rice, corn, vegetables, pest surveillance and forecasting, plant quarantine, pesticide chemistry and toxicology, and training and extension. Thus, the center's existence has been strengthened to provide solutions to the country's pest problems by developing, adopting and extending appropriate pest management technologies to farmers, and formulating appropriate recommendations for use by government policymakers. An advisory board spearheaded by the Department of Agriculture served as guide in pursuing the goals of the center. After 12 years of clustering, the UP Board of Regents approved the restructuring of the College of Agriculture, renamed it as College of Agriculture and Food Science, and dissolved all clustered units and institutes to form independent units, again including NCPC. Today, the center rises up as a center of excellence in pest management with its new leadership and experts that would continue the works and realize the vision of world-class excellence in crop protection and pest management. The center stands alongside premier UPLB institutions at the Agricultural Science Park of the University of the Philippines, Los Baños. NCPC is headed by a director, deputy director, and division heads, supported by a number of research and extension staff, and backed up by administrative personnel. Unified by the standing goal to support agriculture programs to ensure food security, environmental sustainability, and the upliftment of the general public's quality of life. The staff work together harmoniously with each other and devotedly pursuing productivity. Researchers occupy laboratories with cutting-edge equipment to support pest management researches in the fields of plant biosecurity, pest management, pesticide management, and public service. Extension services are also active components of public service. Technical services are focused on plant health clinic, the quick response team wherein NCPC addresses serious pest problems through quick response to pest outbreaks. The center also caters to pesticide residue analysis and pest database management. NCPC leads in national research and development programs with solid international and local partnerships, regional crop protection centers, Department of Agriculture regional field offices, Bureau of Plant Industry. The center's advisory board members that continue to provide guidance in the performance of its RDE functions are respected and well-accomplished institutions. NCPC strengthened and dynamic researches, human resources, and multi-institutional collaboration continue to meet the needs of various stakeholders, including the academe, LGUs, farmers, policymakers, and other government institutions. Welcome to the National Crop Protection Center.
Okay, isang napakagandang umaga po sa ating lahat. Sa mga kasama po natin dito sa Zoom at sa mga nanonood via Facebook and YouTube Live. Good morning po. Ako po si Charlotte Agusos mula sa National Crop Protection Center. At kami po mula sa NCPC ay nalulugod na kasama namin kayo sa pasimula ng week-long celebration ng aming 46th anniversary. So we begin our celebration with the anniversary webinar series on the futures of crop pest management. This series aims to discuss the trends, the innovations, and emerging issues on crop pest management. Ngayong umaga, ang topic po natin is on Foresight for crop pest management in research. Ngunit bago po tayo magsimula, narito po muna ang ating house rules. Okay, sa atin pong participants dito sa Zoom, please keep your microphones on mute to minimize disruptions. Kung kayo po ay may katanungan, pwede niyo po ito isend via Zoom chat box o kaya ay sa Facebook or YouTube comment section. Later tayo po ay may open forum kung saan tatalakayin natin ang inyong mga katanungan. And now, to formally begin our program, let us call on the Director of the National Crop Protection Center, Dr. Barbara L. Kawili.
Salamat, Char. To our resource speaker and the Executive Director of the International Service for Acquisition of Biotech Applications Incorporated, Dr. Rodora Romero Aldemita. To our colleagues at the University of the Philippines, Los Baños, and to all our participants from other SUCs and institutions, ladies and gentlemen, a pleasant day to all of you. Today, we begin the week-long 46th anniversary celebration of the National Crop Protection Center with a webinar that will give us an idea about what the future of crop protection and disease management in research will be to address food security and sustainability. With climate change, proliferation of agricultural technology innovations, and shifts in plant pests and diseases trends, we can say that how we manage crop pests today might differ from how we will manage them in the future. So research plays a very crucial role in ensuring that we all are well prepared for whatever the future in agriculture may be. It is crucial in obtaining relevant insights, in equipping us to handle upcoming risks and challenges, and in addressing the needs of the next generation. So as we listen to today's discussion, it is my hope that we gain vital information about the emerging research-based crop protection practices and technologies. May the discussion also give us hope in our goals of ending hunger, achieving food security, and promoting sustainable agriculture. So with that, I wish an insightful and meaningful webinar. Maraming salamat po. Again, magandang umaga at Hiraya Manawari. Char?
Thank you very much po, Ma'am Bambi. Okay, so ngayon naman po, we would like to request lahat ng ating participants dito sa Zoom to please turn on your video for the photo opportunity. Okay, let's just wait for everybody else. Okay, so first panel, 1, 2, 3, smile. Second panel, 1, 2, 3, smile. Okay, and last panel, 1, 2, 3, smile. Okay, so thank you very much po. Okay, marami pong salamat. At this juncture, nais ko na pong ipakilala ang ating resource speaker for today's webinar. She is the Executive Director of the International Service for the Acquisition of Biotech Applications Incorporated, or ISA. She's also the director of ISA Global Knowledge Center on Biotechnology, based in the International Rice Research Institute. She's a member of the DA Biotech Steering Committee, a member of the Technical Advisory Team of the Applied Biotechnology Committee of the DA Biotech Program Office, a board member of the Biotechnology Coalition of the Philippines, the editor-in-chief of the Philippine Journal of Crop Science, and the scientific reviewer of 6 scientific journals. She holds a PhD in botany from Purdue University in the US and did her postdoctoral fellowship at Albert Ludwigs University in Germany. She has served as Chief Science Research Specialist and Biotechnology Coordinator at the Philippine Rice Research Institute and was also a researcher at IRRI. Her research interests focused on genetic improvement of rice through genetic engineering. She has also been a member of the Golden Rice Project team at AUF and at PhilRice. She also has numerous awards. Among them are the Outstanding Young Scientist Award given by the National Academy of Science and Technology, the NAST and Italy-based the World Academy of Science Science Prize in Biology, the 10 Outstanding Women in the Nation's Service Award from Towns Foundation, and the UPLB CAF's Distinguished Award for Excellence in International Service and Cooperation. Ladies and gentlemen, to talk about Foresight for crop pest management and research, here is Dr. Rodora Romero Aldemita.
Hi, good morning, everyone. You want to continue this stuff? Yeah. I'm just reading yung ano. I'm very happy to be here at your 46th NCPC anniversary, and thank you for the invitation, especially to Dr. Barbara Kawili, whom I have been friends with because of her proposal at the DA Biotech Program Office. And we have been connecting through this project, and I hope it's already done. Is it done? Okay, but before we proceed, I'm just going to, where is this? Let's play from start. Okay, yeah. So what I'm going to provide you is a lot of information on what's going on in crop pest management through the use of modern biotechnology. The foresight, the item foresight would depend on what's going on because of climate change. And I might not be able to provide all of the foresight, but we will see what are the things we can do to work to provide information on how do we solution or solve, provide solution for the foresights. So I'm actually, I had my bachelor's degree at UPLB. I'm a plant pathologist. And then I had courses in entomology, in plant pest management. So, but then I moved to biotechnology, and now I'm a science communicator. So I'd like to tell you that because of this background, I'm able to work on the crop science, CSSP, and I've been the executive, the editor-in-chief for the last 10 years, and I hope that you would publish there too. So to start, this is what we have. Why do we need biotech crops? So we have been provided information on how difficult it is to meet the challenges of high global population, climate change, and this will result to severe food insecurity and undernutrition in low-income countries, especially us, the Philippines, and with more than 820 million still hungry. This is with the data which I got in 2020. So this is not going to meet the SDG 2 goal for zero hunger by 2030. So if we do business as usual, meeting this SDG 2 goal is going to be impossible. So let us now embark into what are the things that we can do, what has happened, and how we can help our global population. So I think most of you have biology courses, and we know that DNA, deoxyribonucleic acid, contains the genetic code of life. So everything from man, man, crops, microorganisms, and fishes, they all contain this DNA code, and they can be interchanged, especially now that the tools are available for that to happen. So with that, I'm presenting to you what are the steps in producing biotech crops. First would be we have to identify the gene. So this is basically how genetic engineering works. So we have the gene. It can be incorporated into plant cells through agrobacterium, tumefacient mediated transformation, or through particle bombardment. We can select those which have been transformed, regenerate them, uh, evaluate in the greenhouse, and then evaluate in the field whether the gene that you have incorporated is there and performing efficiently. And then later on, our proponents will have to undergo some more field testing, multi-locational testing under the umbrella of the TWG, or the Technical Working Group for the crop. Now, each of these steps is governed by regulation, and we are lucky that our scientists in the Philippines have started working on the regulation since 1992. And all of this have to be given a permit before the experiments can be conducted. And when I got back from my studies, I was also subject to this grueling task of being conferred or given the permit to proceed to my research at the Philippine Rice Research Institute. And from way back 1992 up to now, we have a lot of biotechnology applications. This is genetic engineering still that we can do, or we can apply for fighting disease, for increasing yield, for environment-friendly technologies, for safer crops, because we can now use technologies that will not expose our farmers and our consumers to some hazardous chemicals, because our farmers will now be able to use technologies using much lesser potent chemicals to control those biotic stresses. We also have nutritious food. We already know about beta carotene enriched rice, which we have been given permission for food, feed, and processing, and for cultivation in 2021. And then for better industry, because we can use a lot of biomass produced using genetic engineering. So with these technologies from 1992, now I'm saying here 1992 because that was the first time that genetically engineered crops were developed, but the commercialization process, the commercialization per se happened in 1996. So here you will see that crops have been developed to improve with improved characteristics, and that about 190.4 million hectares were planted in 2019. So here you can see the developing countries, which were a bit late in adopting compared to the industrialized countries. And there was here a section where by 2011, the developing countries have moved up. So currently by 2019, there are more countries who planted biotech crops compared to the industrialized countries. And you will see it in the next few slides as I move along. So here, now would like to tell you that most of these crops, the biotech crops, contain herbicide tolerance, insect resistance, disease resistance, which contributed to food sustainability and profitability to the farmers during those more than 25 years. And looking at the graph, you will see that insect resistance and herbicide tolerance were the most planted crops. But as you see, the stock traits, meaning both insect resistance and herbicide tolerance, are present in the varieties. But we can, when we talk about pest management, we think about herbicides, or we think about weeds, we think about insects and diseases. And of course, we also have a lot of small things like mycoplasma and some endogenous living organisms that reside in the plants, which somehow there is symbiosis, but at the end, you will see that it will affect the crops, or the productivity of the crop. But we don't deal in that. In this biotech crops, we only deal with herbicide tolerance, insect resistance, and disease resistance. And also, from 1996 to 2019, the major crops which were given this traits were corn, soybean, canola, and cotton. So those are the 4 crops cultivated very immensely at high hectarage from 1996 to 2019. So now we would like to see how do scientists put their efforts in producing these insect-resistant varieties. So this will cover protection of crops in the field and in the storage. So we have these insect species, Lepidopterans, Coleopterans, Hemipteran, and sometimes we have multiple insect resistance in the varieties or in the events that were produced. And you may look at this. So there were 10 crops which were genetically engineered or genetically modified to have these insect resistant traits. So we have cotton, which has the highest, maize, which has the highest, followed by cotton, and then potato. So we can see how important corn is, because we know that corn is affected by European corn borer, Asian corn borer, rootworms, and other insect pests. So there were 307 genetically engineered events. That means one gene or multiple genes can get into what the insertion site of the chromosomes in the crops, in the genome of the crops. So next one, so we see how important it is to have insect-resistant traits. Next one would be the herbicide-tolerant traits. And here we see different types of herbicides. 2,4-D, dicamba, glufosinate, glyphosate, imazamox. I don't know these herbicides, but from the ISA database, so if you would like to see more detail on this, we have the ISA website. You can look at it and look at the GM approval database, and it will provide you the data that you would like, and it's updated regularly. So we know that I think 40% of all crops in the world are affected by weeds. So you can see here that maize is also the highest one, which has 215 events, followed by cotton as well, and then soybean. But then later on, you will see that soybean has, herbicide tolerant soybean has the most, is the most planted all over the world. So there are more crops also with herbicide tolerant traits. So what happens here is that crops are given resistance to this herbicide, so that during the spraying or during the killing of the weeds, you can just spray with any of these chemicals, and your genetically engineered crops will be standing and your weeds will die. So that's why there is zero cultivation, and we have lesser application of um, hazardous and lethal chemicals. Okay. Now, other biotic stresses. So we have potato foliar leaf blight resistance. We have 4 events. Potato leaf blight disease, 1. Soybean nematode resistance, 1. And this is the viral disease resistance is the most popular. So we have bean golden mosaic virus resistance, which has been approved in Brazil, but still not planted. Papaya ring spot virus resistance. This is basically planted in the US and in China in large hectareages. Plum pox mosaic virus, this has been approved, but is not yet on the ground. Potato virus Y, leaf roll virus, it's still there, it's being planted. Squash with multiple virus resistance is there in the US. Sweet pepper, cucumber mosaic virus resistance, and tomato, cucumber mosaic virus resistance. And there are still other crops being improved through genetic engineering um, in other parts of the world. So with these types of genetic engineering or improvement in crops, I would like to tell you also that besides this biotic stress tolerance, we also include some consumer traits that are incorporated in some of these varieties. So that's why we have stock traits, not only, for example, for example, we have insect resistance with drought tolerance, with sweet, or how do you call this, with high sugar content in corn. So that's how stacking of genes is achieved. Now we can have, through the biotech crops, we have contributed to food security, sustainability, and climate change. And I would like to emphasize here that the productivity, the providing a better environment because of the decreased use of crop protection products. And the crop protection products, of course, there is going to be a strategy where, for example, the crop is already protected through genetic engineering against lepidopterans, but then it's not protected from the other insects. So that's when other crop protection products can be used, but then we can also apply IPM, where farmers will learn how much and which the better pesticide should be used. So that's why in biotechnology, we marry both the not the organic, the genetically engineered, and then the conventional protection that we use. We cannot, some of the products the farmers can use should not be hazardous. So that's why it is important that farmers should be taught how to use these products. So on top of this, providing a better environment, we can conserve biodiversity, we can reduce carbon dioxide emission, and we can help alleviate poverty and hunger. This is a data for 2019, and we're still producing the 2020 data. And we are hopeful that, and we're looking forward that the crop productivity will still continue using genetically engineered crops. Now, there are still a lot of activities in genetic engineering to protect our crops from these biotic stresses. But then there are more technologies that came out. Now, I'm just going to tell you that this is what is going on in the Philippines. So we have crops approved for import. We have food, feed, and processing. We have alfalfa, canola, cotton, eggplant, and others. And this is how biotech maize looks like. So this one is the non-GM corn. As soon as the Asian corn borer eats or bores into the corn cob, there are, of course, wounds that are opened up, and these anthracnose aspergillus come in and infect our corn, which produces aflatoxin and which is harmful to our livestock and also to humans if you eat aflatoxin because it is carcinogenic. So compared to the one on the right, which is very clean and no holes and no black aspergillus in it. So biotic maize has been planted by 673,000 farmers in the Philippines in 875,000 hectares in 2019. So you can see here the benefits, and this has been published by Graham Brooks in 2021. You can see that this has been increasing, of course, because our increasing hectareage but in planting biotech maize. So the upcoming Pinoy products, Philippine biotech products, we have golden rice, of course, you know that. We have beta carotene enriched rice. So compared to the white rice, so it can alleviate the vitamin A deficiency in our coast people who are living in the coastline who are very susceptible to vitamin A deficiency, and also people in the uplands who do not have enough supply of vitamin A source. We also have BT eggplant, which has been given approval for food, feed, and processing. And then they are now looking at propagation approvals very soon. And then we have BT cotton, which has the same strategy where we have the Cry1A gene. And this is against the BT cotton, against the cotton bollworm. This is also under multi-locational testing currently. So we're hopeful that these biotech will be coming one after the other. So plant science is constantly evolving, and because of when I say earlier on that business as usual will not solve the problems of hunger and poverty, and of course we have the climate change problems. So here in the beginning of 2000, gene editing has been very much researched. There are several ways by which gene editing can be conducted. And I'm not going to involve you into the details of the technology, but suffice it to say that it can deactivate an unfavorable characteristic by just cutting and splicing out the genetic sequence that you don't like, and then it can enable a beneficial characteristic. So this is one way where you can just target it. In genetic modification, the modification happens randomly. So you won't know where it goes. You will know it after you check on it. But for gene editing, you will devise ways by which you will make sure that the deactivation or the splicing can be directed to where you want it to go because of the sequences. And of course, they design the vector so that it will direct them to the correct insertion or the cutting site. Now let's look at how this happens. So I'm just sharing you the slides by Dr. Gabby Romero. So this slide, it's similar to a word processor, find and replace. So you will find it and replace the function. Gene editing tools can delete or edit nucleotides within a specific gene to achieve a desired or improved characteristic. So you can have deletion or loss of function. So you will just delete a single base or a few bases, and then so that's when you just have a deletion and you will have a new function. Now you can also replace where you can change in function. So after you cut, you will replace a few sequences with a primer, and then you will have a new function. So we have SDN1 and SDN2. Now SDN3 is when you cut and replace the gene where the gene comes from another organism. It can be the same species or it can be a different species, and it's just like a genetic engineering. So I hope you got that. We can discuss that later. some more. So we can enable a beneficial characteristic like a drought tolerance. We can deactivate an unfavorable characteristic, and we can break genetic links. And because of this technology, we have gene editing tools hold promise for farmers, consumers, and our planet. So we have the farmers, another tool that improves the pace and scope of innovation to deliver better seed products. And mind you, it is faster, much cheaper, and of course, it will get to the farmers much faster. Consumers and agriculture system that meet society's needs. I'm going to describe to you some of the new products which target the consumer needs, and then our planet, because we are conserving our planet by using by smarter use of inputs like pesticides and natural resources. So we have currently potential applications in weed management, disease resistance, drought tolerance, quality improvement, and enhanced nutrition. So I would just like to invite you to come to our website. We have the ISA website, and you can look at the genome editing resource, and it contains all of the articles published from 2016 up to now on genome editing, and it's now categorized into many different categories. And we have also published a primer in genome editing, and you can download it for free. It was published last December 2021, and currently we have more than 2,000 downloads. So I invite you to read it. It's written in very laymanized form, and experts from the Philippines, from Africa, from Argentina, Singapore, have written their pieces on genome editing. So I'd like to introduce to you to the first genome edited product in the market, which is soybean. So soybean, it's the market name is Calyx. So it cuts off the pathway from oleic acid to produce linoleic acid. That's why it's high oleic acid soybean. And it's now available in the US, and I think it's also available in the Philippines. I saw some products already in the shelves. The second one is for GABA tomato, which is approved for commercialization in Japan. And it was approved in 2021, as you can see there, and it enhances blood pressure lowering. It's already available in the market, launched in May 2021, released in 2021, and you can already buy it through online, and the seedlings will come to you when you're in Japan. I don't know when it's going to be in the Philippines. So here are the gene edited crops in the pipeline. Of course, the first thing that our scientists do is to target yield. And so first one would be these are researches that were published in our crop biotech update in the last 2 years. So I just got some of the examples for your appreciation on what's happening with genome editing and how exciting it is to get involved and know what is coming to us. So we have camelina, which has 55% increase in oil, and it is now going to be commercialized in the US and Canada. It's already commercialized in Argentina, and it is not regulated. We have canola, which has shorter to minimize lodging. It's highly branched, meaning more pods, so it's easier to harvest, and it's already in the last leg of the pipeline in Canada. Now let's go to crops which have a disease resistance trait. So we have pepper with resistance to pepper banal mottle virus. This is in France. Wheat, which is resistant to powdery mildew in China. Banana, which is a staple in Africa. So we have resistance to banana santumon as well. In the US as well, the resistance to white mold of canola. We also have a biotic stress tolerance in maize. So we have drought resistance and with lower lignin content for better digestibility as silage. This is in Netherlands. We have barley with delayed germination, which prevents pre-harvest sprouting due to unexpected rains. So this is in Japan. Oh, okay. So this is what I'm telling you about the consumer traits. Since most of our, we would like to feed the growing population and that food can be accessible, but at the same time, it can be nutritious and it can be attractive to people. So first one would be sorghum. And this is, they have fragrance traits in it for food, liquor, and vinegar brewing. This is being developed in China. Potato with decreased cold-induced sweetening and acrylamide formation in fried potato. So this is better for our health and it prevents food wastage. We also have something like this, genetically engineered one by what is that? Simplot company in the US, but this one is through genome editing, and this is being developed in Australia. Then we have tomato, which increased sugar content by 30% with no effect on fruit size. This is in Japan. And chicory, the source of natural sweeteners, inulin. So it no longer contains the bitter compounds through genome editing, making the processing cheaper, easier, and more sustainable. This is in Netherlands. Wheat, which has reduced asparagine and eventually the carcinogenic acrylamide during baking. This now in the UK. And leafy greens, which have nutrient-dense salads with rich greens and colors. So you know that with rich greens and colors, we can get more nutritious leafy greens. And they are also embarking into seedless berries like blackberries, black raspberries, and seedless cherries. Okay, so in the Philippines, what do we have? Our Institute of Plant Breeding is doing high lycopene tomato and low phytate corn through genome editing. Bell Rice is conducting optimal amylose content in rice. And at the International Rice Research Institute, we have bacterial blight resistant rice, fungal resistance, and in improving yield. Now, I cannot help but provide you information on what's happening in other species other than crops. So we have genetically genome edited fishes, which have been approved for consumption and release in the market in Japan. So this is sea bream with myostatin protein knocked out to increase growth rate. So our sea bream fishes can now mature in lesser time. And then puffer fish, if you're familiar with puffer fish, it has disrupted leptin gene, it gained appetite and weight in such shorter time. So if you may remember, Japan has the highest, I'm just telling you that Japan has the highest approvals for genetically engineered crops, but they have not commercialized genetically engineered crops. But they have embraced genome edited crops. So they are now one of the first countries who have commercialized genome edited tomato. And now we have genome edited fish, which they have just approved for commercialization. And there are more fishes and crops in the pipeline for commercialization purposes. Now in animals, so this is now USDA, we have a slick hair coat in cattle, which will regulate their temperature. So this is another way of these animals to get, to adapt to climate change, which will bring enormous high temperature increases. And then we have a typical horn. We have here, cows always have horns, but then they were able to genome edit it so that they can have now hornless cow. This is in the US and also in Australia. And you know why it is important for them, for the humans as well as for the animal, for the cattle, so they don't have horns. Because to dehorn the cow, there is the pain and the the probability that they will get infected in the wound. And then of course, with horns, it's hazardous to humans when they approach the cows. So that's why that they tried to develop these cows without horns. And then there is a lot of applications now in healthcare. We can produce complex antibiotics to reprogram pathways to future medicines, and which is urgently needed currently. And we know that through genome editing and through genetic engineering, we have produced vaccine, the mRNA vaccine of Pfizer and Moderna and Janssen. So through biotechnology, we were able to produce those vaccines immediately, and they were able to see that it really fights the disease. And now this is a bird's eye view of what's going on in terms of regulating landscape for SDN1 gene. This is when the gene is knocked out and just spliced together. So these are the blue countries, are countries which have approved SDN1 edited products as conventional products. So they are not regulated at all. Red, these are countries which regulate genome edited crops as GM. So you can see here it's Europe and South Africa. And yellow, where there are noticeable policy making decisions. And well, the Philippines has already released its new breeding innovation. No, it's going to release its new breeding regulation products guidelines within the month, I think. And we're looking at similar guidelines as the Latin American and US ones, following the Argentina pathway. And these are within the year 2022, we have seen that China, Nigeria, the UK, Japan, and Canada are improving their regulations so that gene edited crops cannot or can be given minimal regulation or assessment, so that the products, the gene edited products, can be given or can be provided and be distributed to farmers for distribution, for planting and consumption. Now, there have been studies on genome edited crops so this is the study in Germany where consumer preferences were studied, and majority of the 32 survey participants showed willingness to buy CRISPR tomatoes or genome edited tomatoes. In China, the same way is true, that consumers feel fear of novel food technologies can be minimized if they know the science. And then in Korea, the same way is true, with 20 to 50 years old respondents prefer genome edited products over GM, and they respond better to novel technologies based on their scientific knowledge. So it's important that we provide the scientific knowledge on the new technologies. So in communicating about these products, we have to highlight the objective information about genome editing must be provided at the government and private levels when actively introducing the novel technology. cite that a process of education and promotion about the difference between GM and genome editing are necessary. And it is important to have appropriate risk communication and dissemination information in the private and the public domain. Looking at the gene editing market projections, because of its application in crop protection, in health, to various organisms, you will see that there's a forecast for an increased market of genome edited crops till 2022. So this is about 2028, which is about $19.45 billion. And this is my last slide. So looking at this slide, we can see the very, how to call, lagging. It's very slow increase in productivity. But due to molecular breeding, no, we start with biotech trend and molecular breeding. We're looking at 18 tons per hectare increase in corn. This is only in corn. How much more in the other crops? That we can attain this much increase if we use the proper technology, which is modern biotechnology. So when I say that we look at the foresight, so with the biotic stresses that we are encountering right now, because of climate change, we're expecting more of these new pests as the climate changes. So we have to be ready. We have to equip ourselves. We have to know the technology, what's present around us in the global community, global scientific community. We can partner with those universities and countries which have the products. So we don't need to start from scratch. So we can have collaborations, and then we can produce our own through those collaborations. And we hope that you learned something from my talk, and I'll be ready for any questions. So I'll just stop share right now. Thank you.
Okay, so thank you very much po, Dr. Aldimita, for that very informative and very interesting discussion. So with all the changes that we are experiencing in the world, as our resource person said, it should not be business as usual. So we have these new products, new technologies with so much potential, and indeed that is very exciting. Okay, so now let's move on to the open forum. I would like to call on my colleague from NCPC, a university researcher and our research coordinator, Dr. Marita S. Pinil.
Hello, good morning, everyone. Thank you, Shar, and thank you, Dr. Aldemita, for sharing with us the global status and challenges of using biotechnology and how the modern biotechnology, like gene editing, will address those challenges to attain our global campaign on zero hunger. Okay, at this point, we have an overwhelming number of participants from different organizations. So we're expecting a lot of questions. And for those who would like to ask their question, kindly use the raise hand button. And then once I called you, kindly state your name and affiliation to be recognized. And sa mga medyo nahihiya po dyan, you may type in your questions sa ating chat box and I will read it for you. Okay. Okay, meron agad tayong buwan naman ng question. Okay, Ma'am Karen, please.
Hi, ma'am. Good morning. This is Karen po from the Institute of Weed Science, Entomology, and Plant Pathology. Ma'am, hindi niyo po nabanggit ito sa presentation, pero I'm just interested on the status of the application of BT talong for propagation here in the Philippines. Thank you po.
Thank you for the question. Actually, we have not yet propagated it. We already have the approval for food, feed, and processing. The proponents at the Institute of Plant Breeding are now, I think they have, well, I'm not sure if they have applied for propagation yet, but we have already information that they're putting together all the dossiers so that they can apply for propagation. It's not yet
Thank you,
propagated.
ma'am. Thank you po.
Okay. Okay, Ma'am Bambi, please.
Hi. Ma'am Ola, they have applied for propagation na po.
Already. Okay. Thank you, Bamboo.
Okay, some more questions? Okay, Ma'am Ola, just ano po, point of clarification lang para sa mga hindi naman po researchers or those who are not engaged naman sa biotechnology. Do we consider genome-edited crops as GM crops also? Or do we have to strictly follow the definition?
Is this Philippine context?
Yes.
Yeah, okay. Sa Philippine context, we already have, the draft. So, yun nga, when I said kanina, we are following the Argentina statements that SDN1, which is just you cut and then put it together. You basically silence the undesirable gene, and then you have a new phenotype that will come out. So it's going to be not regulated as GM. For SDN2, I think they're saying that as long as the inserts, because in SDN2 there will be a primer for putting together the gene. So if you have a primer, yeah, so may primer ka dyan. So syempre merong insertion. If the insertion is less than 20 base pairs, then it's still considered as conventional. And then if it's longer, and then the gene that you inserted would be coming from another species, then it will be, or a related species, related or unrelated, then it will be considered as GM. So, and this is, I think, going to be released very soon. I don't want to preempt it. Yeah, baka ako Pero they have, it's coming out very soon. And there was a committee who put together the guidelines, and they are really expert scientists. And we know that the interest of the scientists and the farmers are being put into consideration, and hopefully it will alleviate all of our problems in agriculture. Ayan, thank you.
Okay, thank you, ma'am. We have a question here sa ating chat box. Ma'am, from DMSU, si Ma'am Elizabeth. So, have you started doing gene editing with insects, or will try to explore such in the future? I'm a researcher, sericulture, and interested with gene editing in silkworms.
I have already presented the traits being done in the Philippines by our research institutions, and it doesn't mention insect resistance as yet. And when I provided the information, the global information, and I already made some research, there are upcoming insect resistance traits developed through genome editing. And well, it depends on your target trait. So the first thing that you have to do is to understand the genetic sequence of silkworm and identify where is the gene that you're interested in. If you would like to cut off or to inhibit or to change your phenotype, then the gene that governs that phenotype should be identified, and then you will know what trait or what procedure you would like to do. So it's genome editing is not very simple in the beginning, because you have to make a sequence first. You have to know the genomic sequence of the gene of the organism. At this point, it's silkworm, and then you look at what trait you would want to improve in silkworm. So maybe if you want to improve the production of the silk, then you have to, where is that gene? And then what would you like to do? Do you want to splice off the inhibitors, or would you like to insert inducers so that the silkworm will produce more silk. So that's the strategy. Currently, in the Philippines, we don't have that research yet. I didn't see in my search anything on silkworms yet. But you know, science has no boundaries. It may come out. And if you do this yourself, you will be the first one. And hey, you'll be the first one to publish. That's a good one from the Philippines. Yeah.
Yeah, that's a pioneering research in biotechnology.
Uh-oh.
Thank you, ma'am. Okay, some more question. From Sir E. De Asis. Okay, sir.
Good morning po, ma'am. Ako po si Eric. Isa po akong magsasaka ng palay. Ang tanong ko po ay among po dun sa biotechnological methods or tools na meron po tayo ngayon, ano po yung best method na pwedeng gamitin natin sa palay against both biotic and abiotic stresses? Salamat po.
Okay. So sa rice po, we have heard already that there's development of BT rice somewhere in China. And if you want to have abiotic stresses addressed, then we can put together all of the problems. So currently in the Philippines, we have drought problems, submergence problems, and high temperature problems. In Erie, they have developed some of these varieties already. They have deployed the submergence tolerant varieties, and adding insect resistance to it would be very beneficial to everyone. But we may have to look at what are the ones available in the Philippines, what events or what are the approvals given to biotech rice. To my mind, we don't have it yet, but it's a very good strategy if you would identify what's the problem in your locality. Do you have drought or do you have submergence? Do you have salinity? And then you can look for the varieties which have resistance or tolerance to those traits. And then sometimes our technical working group or our TWUG of rice, they look at the resistance to pests. And that will provide you information on what varieties which have been approved by the National Seed Industry Council that has those abiotic stresses and at the same time resistance to diseases. And they cannot release any variety which has not undergone screening. Our rice technical working group have done, they get information from all over the Philippines on how a candidate variety has responded to different stresses. And they can only release a variety if it has a special characteristics. And you can look at that in their website of rice. I think it's at BPI. Yeah, so I cannot pinpoint you right now, but you have to know first what's your problem in your locality and what are your needs. Thank you.
Okay, thank you, ma'am, and thank you, Sir Eric. So another question, ma'am, from JM Garcia via Facebook. Is using herbicide like glyphosate on glyphosate-tolerant plants can greatly damage our environment?
Actually, glyphosate and glufosinate are the least hazardous. Actually, when you spray that, within 5 days, the active ingredient is already not there. So it kills the plants, the weeds instantly, and it degrades very fast. So it does not contaminate the soil. It's not hazardous to humans who are using it. And even if you spray it to crops, it will not be, there's no residue, herbicide residue on the plant. So glufosinate and glyphosate belongs to the list hazardous chemicals. And there are studies in Australia and in Latin America, I guess, who have been using these herbicides, and they couldn't detect any contamination in their underground water because they are really safe and they degrade fast. Thank you.
Okay, thank you. Ma'am, follow-up question lang dun sa question ni Ma'am Elizabeth Obra kanina. How about collaboration daw po when it comes to gene editing research in silkworms?
Yeah, you can do that. We are not a research institution. ISA is provides service, knowledge service to the global community on biotechnology. You may want to go and ask at the DA Biotech Program Office. If you have a proposal, you can just connect to them, and they are always accepting new proposals on biotechnology. And if there is, if you can, well, if there is an ongoing research in other universities or research institutions abroad, then you can get connected with those research institutions, with the scientists working on silkworms. I remember there is one ongoing research in Vietnam, but they haven't really published it yet. So I don't know if it has materialized or they are producing this genome editing silkworm. I tried to connect with those people in Vietnam last year, but there was no response. So let's see if we can do collaboration. If you, well, yeah, you can submit the proposal at the DA Biotech Program Office. They accept these proposals and maybe connect you to the right contact scientist.
Okay, thank you, ma'am. So, Ma'am Elizabeth, pwede na kayo agad magawa ng proposal nyo and submit it to DA Biotech. Okay, some more questions po? Meron pa po ba? Okay, si Sir Eric po ulit. Okay, Sir Eric.
Ma'am, salamat po. Ma'am, follow up ko lang dun sa question ko kanina. Kung pag-usapan po natin yung outside the context po ng Pilipinas, since hindi pa talaga fully accepted ang gene editing sa Pilipinas, regardless sa crops. So let's say kung sa ibang bansa po, let's say China, ano po sa tingin niyo yung pinaka magandang method na gamitin para po doon sa mga stresses na to? Let's say CRISPR ba, zinc finger ba, or gene knockout ba? Ano po sa tingin niyo yung para sa rice?
Okay. Genome editing tools. Yeah, you have provided, you have identified or enumerated the tools, but currently it's CRISPR. Genome editing is the most popular right now, although we already have Tolerance, which is also good because it doesn't give yung leakage in targeting the genome edits. So yeah, so CRISPR is the best. In rice, I think they're also using, at IRI actually they're using both Talents and CRISPR. Yeah.
Okay, some more questions,
Salamat
Sir
po, ma'am.
Eric?
Okay.
Okay, thank you po. Tanong pa po?
Isa, may nag-raise kanina, I didn't see the name.
Si Sir Eric po ulit. Okay, ma'am, ako pala may tanong. Among the mentioned gene-edited crops, was there any report po so far of the irreversible effect sa mga non-target organisms or even sa consumers?
Irreversible effect. Actually,
I mean, negative effect or
negative effects. Okay, let's look at genetic engineering. Genetic engineering has been there for more than close to 30 years. And we have not seen any, or there is no reported hazardous effects or lethal effects to humans. It's all positive. Actually, mas naging positive pa nga yung there's no hazardous effects. And then for genome editing, we don't have that much data yet because it's the beginning, but looking at the science of it, where it is more targeted than genetic engineering, then the more non-hazardous or more less riskier it is, safer are the genome edited crops. But they are both safe. They will both undergo assessment, risk assessment, except for SDN1 and sometimes SDN2, because looking at the sequence itself will tell you whether there would be any risk in using it. So basically, wala talaga. It's very safe. And of course, scientists will not release anything that will be hazardous to the human population.
Okay. Thank you, ma'am. Follow up lang po, ma'am, doon sa question ko. Ma'am, how about sa tingin niyo yung acceptability ng ating mga Filipino farmers or consumers once na meron na tayong gene-edited crops commercially available?
Yeah. Actually, we're ano to? We have started a perception study in the Philippines, not only with genetic engineering, but also with genome editing for crops and animals. And we will have it very soon, the result within the year. But for genetic engineering, the acceptability of farmers and consumers was high. And of course, they have also identified the sources of information that they would like, that they would prefer, that they would believe in. So we're looking at, for scientists, is the number one source of information that they will believe in. So if the scientists say it's safe, then they believe the scientists, because they know that they are doing their best to make the product safe and as useful, beneficial to the human population. For genome editing, I think there's no question about it, because genome editing is a lot more improved technology than genetic engineering. Although there are some traits which genome editing cannot address. So for example, if actually insect resistance is one of those traits which genetic engineering can address better than genome editing. Because what genome editing does is you have to look at the sequence of the genome, and then you will have to identify which one you would edit. And in crops, the very less occurrence of insect-resistant traits within the genome, or inhibitors for insects, against insects. So scientists prefer using genetic engineering, and also we can stack more genes against insects in genetic engineering compared to genome editing. So that's why when I am asked, do you think genetic engineering will be passé? No, it cannot be, because there are still some other traits which genome editing cannot address, especially the multi-gene stacking. Hindi pwede yun sa genome editing. But of course, there is a genome editing a tool where you would just insert the different genes in the correct site, because in genetic engineering it's always random, but in genome editing you know where to insert it. So it may come that multiple insect resistance genes can be hooked up together in a sequence and then introduced into the plant genome at a specific site. The site where they're not going to be methylated, where they're not going to be spiced off, it's conserved and stuff like that. Nagiging technical na. Pero that's how it's gonna be. Okay, may tanong pa dito.
May pahabol, ma'am. From Julius Cabantug. Tanong po, yung AZ8433DT po ba ay GMO?
Yeah, this is I think this is let me look at that. Where did you get that number? Kasi I have to look at our GM approval database. If it's AZ8433. DT means drought tolerance. Ano? So if it's AZ, let me check that. AZ8433. Anong crop ito? This is GM.
Rice daw po.
Yeah, I see it.
Ah,
Bayer.
Bayer.
No, sorry, Bayer. German kasi yung Bayer eh. Bayer pag English. Minsan napapa-German ako. Yeah, it's Bayer. It's GM. Yeah, it's GM. And it has drought tolerance as well as resistance to bacterial leaf blight.
Okay, ma'am, one last question before we end po. From Norilyn Anzano, ma'am, may I ask if the gene editing is also safe for those beneficial insects to our field? And what are the advantages and disadvantages of using this gene editing to the biology of insects?
Yeah, so this is same with genetically engineered crops, genome edited crops. You may not need to spray, nah? So when you spray insecticide, it kills everything, including the beneficial insects. So if you don't spray at all because you have genome edited crops or you have GM crops, then the more that the beneficial insects will thrive. And there are already studies on this. You may want to consult works by Graham Brooks, by Shilton, and yeah, there are many studies to prove that GM and genome edited crops, which are insect resistant, allow the beneficial insects to thrive.
Okay, thank you ma'am, and maraming pong salamat. Buena mano po namin kayo sa aming 46th anniversary, and thank you for this very fruitful discussion. So I turn it to Ms. Shar.
Okay, so thank you very much po, Dr. Pinili, for facilitating the open forum. Thank you din po sa ating participants for your questions. And of course, thank you very much sa ating resource speaker, Dr. Aldimita. And to show our gratitude, we are awarding Dr. Aldimita a certificate of appreciation from the center. So allow me to read the citation. National Crop Protection Center, College of Agriculture and Food Science, University of the Philippines, Las Bañas. Certificate of Appreciation is awarded to Dr. Rodora Romero Aldemita for serving as the resource person during the NCPC Anniversary Webinar Series episode on the topic Foresight for Crop Pest Management and Research, held on 16 May 2022 via Zoom. Given the 16th day of May 2022 at the National Crop Protection Center, College of Agriculture and Food Science, University of the Philippines, Las Bañas, Laguna, Philippines. Signed by yours truly, Chair of the Seminar Series Committee, and Dr. Barbara L. Kawili, NCPC Director. Muli, marami pong salamat, Dr. Aldemita.
Thank you so much for having me.
At ang ating pong participants ay makakatanggap din ng kanilang Certificate of Attendance sa webinar na ito. Mangyari lamang po na sagutan ng evaluation forms sa bit.ly/AWS-Eval1. Amin pong ipapadala via email ang inyong certificates. We'll also provide this link in the Zoom chat box and sa Facebook and YouTube comment sections. Okay, at this point, to close our program, let us have the Deputy Director of NCPC, Ms. April N. Alviar.
Thank you, Char. To our resource speaker, Dr. Rodora Romero Aldimita, and to all our attendees for today's webinar, a blessed morning to all of you. As we end the first episode of our anniversary webinar series, I wish to thank Dr. Aldimita for her very informative talk about biotechnology, which featured gene editing and its application on agriculture. This has been a very great opportunity for us to see the potential of gene editing for increasing yield, promoting pest and disease resistance, and advancing sustainability. Truly, we hope that in the future, our research endeavors will be able to address the many challenges in crop pest management and disease resistance and advancing sustainability. Truly, we hope that in the future, Our research endeavors will be able to address the many challenges in crop pest management and agriculture in general that are brought about by the changes in the world. We also hope that through these endeavors, we can make significant strides towards the attainment of sustainable development goals, particularly on zero hunger. I would like to acknowledge the effort made by our webinar team headed by Ms. Charlotte Hagosohos for making this event to fulfillment. More than that, thank you to all of you for active participation in this webinar, especially during the discussion. I hope that the knowledge we have gained and the thoughts we have raised will be useful in our continuing education and future undertakings. To end my message, I would like to invite all of you again tomorrow as we talk about the futures of crop pest management in extension and policy making. Once again, good morning and keep safe, everyone.
Thank you very much, Miss April. At nice din po naming pasalamatan ng aming mga sponsors sa week-long anniversary celebration ng NCPC. So thank you very much to AVC Chemical Corporation, Chemistar Corporation, East Agricultural Products Corporation, Monarch Agricultural Products Incorporated, TMCP Foundation Incorporated, Plants Products, Radisson Agrochemical Corporation, Rainbow Agro, Syngenta, and UPL Incorporated. Salamat din po sa Southeast Asian Regional Center for Graduate Study and Research in Agriculture. And from UPLB, thank you po sa Dairy Training and Research Institute, Institute of Crop Science, Institute of Animal Science, Institute of Weed Science, Entomology and Plant Pathology, Information Technology Center, Office of the Vice Chancellor for Planning and Development, and Office of Public Relations. So thank you for helping us have a meaningful anniversary celebration. At kayo pong lahat ay aming iniimbitahan bukas for the second episode of the NCPC Anniversary Webinar Series 2022. So we will have Dr. Rex Navarro, Senior Technical Advisor of the Department of Agriculture, Bureau of Agricultural Research, to discuss foresight for crop pest management in extension and policymaking. So you can register at bit.ly/2022AWS2. Yes, at manatili po kayong updated sa aming mga activities by following us on Facebook, checking out our website, and subscribing to our YouTube channel. With that, we wish everyone a great day ahead. Let us end our program with the singing of the CAPS hymn.
Maraming pong salamat, Ma'am Ola.
Welcome, Mami. See you soon.
Thank you po.
Face to face. Bye.
Thank you po, ma'am.
All right, bye. Leave na ako, ha?
Yes, ma'am.
Ano bang date ng Friday?
Friday 13. Friday 13.
Ano bang pangalan ng director ni ano?